Biotechnology. This is a broad field which involves two kinds of studies. First of all, the principles and the processes which govern the biotechnological techniques, instruments, tools which involves various principles in which biotechnology works and various processes that are involved in this technique. And the second is its applications. Now, let us know what biotechnology really is. So, this basically is a technique that involves the use of living organisms, their cellular components, and their enzymes to produce useful products and processes for man and mankind as a whole. The term biotechnology was coined by Carl Erecke. According to European Federation of Biotechnology, it is the integration of natural science, cells, parts, and molecular analogues for products and services. There are two phases of development of biotechnology, the old or traditional and the modern biotechnology. The old traditional biotechnology. It is also known as conventional technology. Conventional means it has been in use since time being, and that is why it is also known as conventional. The production of curd, bread, wine, alcoholic beverages, cheese, all of these have been produced using the traditional biotechnological techniques. Now if we deal with the modern biotechnology, this is a new branch because it has been developed during the 1970s and afterwards. So this basically involves the development of highly new and useful traits in crop varieties and animal traits by the use of genetic engineering. So by using genetic engineering, useful and desirable traits can be produced in animal breeds and crop varieties. Example of modern biotechnology is in vitro fertilization, which leads to test tube baby, and also synthesis of a new gene and using of that. Another example is development of DNA vaccine or correction of defective gene. Now, if we talk about the principles of biotechnology, that is what forms the basis of this technique. So, first of all, there is genetic engineering. In this technique, the chemistry of the genetic material is altered by introducing some desirable traits and some manipulations which are desirable for us. So we introduce this manipulated genetic material into the host organism, and then this altered genetic material alters the phenotype of the host organism. So this basically is known as recombinant DNA technology or genetic engineering. Second principle is that we have to maintain a microbial contamination free, that is a sterile surrounding in chemical engineering. Whenever we are making some kind of manipulations, we just need to have desirable characters or traits. So we need to prevent any kind of contamination so that desired product grows and there is no undesirable change on what we are planning to get. So this sterile surroundings only helps to grow the desired organisms or even the eukaryotes in large quantities. This basically helps in the manufacture of antibiotics, vaccines, enzymes, medicines, hormones, and so on. This is used by the various industries for the production of various antibiotics, drugs, vaccines, etc. Now comes the application of biotechnology. Biotechnology plays an important part in many fields such as agriculture, medicine, transgenic animals, ethical issues. So the first application is agriculture. Why we need to know more towards the agriculture in biotechnology? The answer is increase in the need towards food production. Even though the Green Revolution has increased the supply of food thrice, It could not possibly afford all the people because the population was increasing tremendously. So the farmers had to rely on the chemicals in order to increase the productivity. But there was a problem because these chemicals were too expensive for the farmers to afford, so they have moved towards the organic culture. In this, the conventional way of breeding doesn't yield the enough results, so they need less expensive and at the same time an effective way of producing food products. So finally, genetically engineered crops produce the result that they expected. GMO, that is genetically modified organism. It is done by manipulating genes of plants, animals, fungi and bacteria, generally through a method of recombinant DNA technology. So the advantages which we have with the help of these organisms are environmental resistance, developing pest-resistant crops, reduce pest harvest loss. Through this, we can actually delay the ripening process of a fruit so that it can withstand more days, increase efficiency of minerals and enhanced nutrition. Example of this is vitamin A enriched rice. The second application of biotechnology is in medicine, genetically engineered insulin. For the insulin patients, there will be reduced or stopped secretion of insulin in their pancreatic cell. So the alternate remedy could be obtaining insulin from the animals like pig or rat, but these kinds of insulin produced from animals will not suit the human body. So what is the actual form of insulin in human body will be chain A and chain B, which is linked together with disulfide bond along with the C peptide bond. But after this process, the pro-hormone status is processed to form an insulin hormone, that is a peptide bond is break from chain A and chain B, and this kind of process can be enabled with recombinant DNA technology, that chain A and chain B are formed separately and are extracted and finally binded with the help of a disulfide bond. The second application is gene therapy. In this, the correction of gene defect is diagnosed in the early stage. And the third is molecular diagnosis. The third application is transgenic animals. Transgenic animals can yield variety of advantages to the society. First one is normal physiology and development. In this, we can know how the genes are regulated and how it affects the normal functioning of human body. Second one is study of disease. We can know how the genes are contributing to the development of diseases. So the third one is biological production. With the help of these transgenic animals, we can actually develop products that can help human beings. Vaccine safety. This is mainly done to test the safety of vaccines such as polio. And the fifth one is chemical testing. Then comes the ethical issues. Indian government has formed a board known as GEAC, Genetically Engineered Approval Committee, and their main role is to make decisions for the genetically modified research and safety of introducing the genetically modified organisms into public services. And the next one is biopiracy. Industrial biotech is helping to fight global warming as an alternative and safer form of global energy instead of diminishing and volatile fossil fuels. It has led to significant reductions in greenhouse gas emissions with the aim to reduce a 2.5 billion tons of carbon dioxide equivalent per year by 2030. Biotech is one of the most exciting sectors at the moment. No other sector has the same promise of extraordinary rewards for investors as biotech stocks, to say nothing of the patients who will benefit from the new drugs and treatments that are developed. With such a broad spectrum of companies and roles within the sector from manufacturing to development to regulatory to commercial release, if you are looking for a long-term career, then you can't go far wrong with a move into or developing your career within biotech. The future of biotechnology is strong. We envision a day when breakthrough drugs leads to a world without cancer or AIDS, a world where there is sustainable development that will tackle energy, food and environmental needs without compromising the Earth's resources or its future.